Welcome, welcome. This is the annual non-coding DNA GWAS primer, and my name is Manolis Kelis. I'm delighted to be here. So the primer today is about understanding non-coding DNA and its association with complex traits. So you've all seen this plot throughout the primers so far, and this is a Manhattan plot. What this tells you is for each one of about 6 million SNPs scattered across the genome, what is the probability with which that genetic variant is randomly associated with a disease? And here, it's minus log 10 p-values, and that basically says that the genetic variants sitting in the FTO locus have a 10 to the minus 60 probability of being randomly associated, which basically means that they're pretty real, that there's something causal going on there between a genetic variant that I inherit when I'm born, and a phenotypic difference in my body mass index, which is a quantitative measure of obesity. The goal of genetic studies is to uncover, in an unbiased way, disease mechanism in order to enable the discovery of new pharmaceutical target genes so that we can enable new therapeutics and ultimately enable personalized medicine. This is very much the goal, this is very much the vision, and this remains the target of why we're doing all of this mapping of the human genome, mapping of genetic variation, association of that genetic variation with disease, and so on and so forth. But this also comes with a huge challenge. When you look in this locus associated with obesity here that reigns supreme above all other loci, you don't find a single SNP that drives the disease, and by golly, here's the mechanism, here's the target gene. Instead, what you see is that there are dozens of genetic variants, all of which are co-inherited. They either come together as a group with a risk version, or they come together as a group with a non-risk version. This is what linkage disequilibrium looks like. And this is the typical depiction of it, of the correlation structure of how these SNPs are inherited through the human population, due to largely their small number of recombination hotspots, a small number of breakings of these chromosomes during meiosis and across evolutionary time. That's the first problem, the fact that there's a lot of them coming together, and that is, something that we've known about for a while. What we did not expect is that when you look across these genetic variants, in 93% of cases, you don't find a protein disruption. So people have always said, oh, we're going to find the gene for this, and the gene for that, and the gene for diabetes, and the gene for cancer, and so on and so forth. First of all, it's not a single gene. It's like hundreds, probably thousands of genes. Number two, it's not genes. It's not the proteins that are affected. It is something outside the proteins. And that's why we're having this primer, because that something outside is most likely the control circuitry of the genome. It's basically how the genome controls the expression of all of these molecules, protein-coding genes, non-coding genes, microRNAs, all kinds of other stuff that is getting controlled by a complex circuitry that is encoded within the same piece of DNA. In this particular example, out of 89 common variants in this locus, all of which are hugely significantly associated with the disease, none of them perturbs the FTO protein. All of them sit within the first and a little bit of the second intron of the FTO gene, but they don't perturb the protein directly. So people very naturally assumed that they must control something about the expression of the FTO gene. And they said, oh, good, at least there's only one gene in the region of association. Because sometimes there's 5 genes in the region of association, and you say, well, we don't know which one. But here's a little secret. FTO turns out to have nothing to do with obesity. It's not the target gene. And yet there's only one gene sitting here. So you're all thinking, well, maybe there's another gene hiding there. No, the true target genes are actually sitting 1.2 million nucleotides away, several genes over, and another 500,000 nucleotides away, again, several genes over. So what's going on here? What's going on is that when you have non-coding variation, you cannot possibly assume that this is the target gene, because the genome is regulated in extremely complex ways. And a genetic variant that disrupts a regulatory element here might sure control this gene, but it might actually control some other gene far, far away. And we should not start developing molecules for just what's under the lamppost. We should really be figuring out what is the circuitry of this region before we decide what to target therapeutically. Everybody with me on this one? So if you understood this part, you guys are golden. So this is it. This is the hard part. It's the circuitry. It's not the genes. So the target gene is not known. The other problem is that the causal variant is not known. So when you have one of these variants that disrupts a protein, we usually make the simplifying assumption, sometimes wrong, that that is likely the causal variant because it disrupts the protein. But when you don't have any of them disrupting the protein, you don't know whether the causal variant is this one or that one or that one. And that's a problem. Why? Because depending on if it's this one, or that one, the corresponding region might be active in liver, or in heart, or in brain, or in lung, or in adipose, and so on and so forth. So understanding the causal variant is extremely important in basically figuring out where these variants are acting. Because the epigenome is extremely dynamic. These control elements are extremely dynamic across cell types, and not all of them act in the same tissues. So not knowing the causal variant means we don't know the cell type of action. And of course, not knowing the target gene means we don't know the relevant pathways. We don't know the mechanism, okay? So that's the challenge. The promise is very clear. The challenge, hopefully, is very clear. So we either stop the lecture here and say there's no solution, or guess what? There's a solution. So the solution is to actually go and systematically understand every last little nucleotide of the human genome. Okay? Turns out it's actually simpler to understand every nucleotide than to understand just the common variants one after the other. Okay? So what the NIH did is invest heavily in not just sequencing the entire human genome, but in actually understanding the entire human genome. How? By funding these large-scale community projects like ENCODE, the Encyclopedia of DNA Elements, or Roadmap Epigenomics Project, or the Blueprint Project in Europe, and so on and so forth. And all of these are working together to basically provide a map, a reference map of human non-coding DNA. And I've been very fortunate to be involved and sometimes lead several of these efforts and contribute to our understanding of the human epigenome and its role in health and disease. Once we can map these non-coding elements, we can start linking them to the control regulators upstream of them and the target gene that they control, and we can start elucidating how these change in response to genetic variation across individuals and how that change contributes to disease. So what we would like to have is for every region of genetic association, understand where it acts, what are the target genes, what are the causal variants, who are the upstream regulators, what are the relevant pathways, and what are the intermediate phenotypes. So what I'm going to tell you today is the work going on in my lab and in several amazing collaborations for elucidating that. So the key insight and the key tool that we're using is that we're trying to bridge this gap between genetic variation and disease variation using something known as the epigenome. How many of you guys have heard what the epigenome is? Raise your hands. Awesome, great. How many have not? Well, I like to think of the epigenome as the great integrator because, number one, it integrates information from genetic differences. Number two, it integrates information from disease differences. The disease itself affects the epigenome and can be reflected in your epigenome. Environmental variables, such as your age, your sex, and other, comorbidities, affect your epigenome. And the epigenome interacts with a lot of other transcriptional processes, microbiome differences, immune infections, behavioral factors, and so on and so forth. So the epigenome sits in this very central place between genetic variation, environmental variation, disease variation, and all of these covariates. That's why we love to study it. So how do we study this, and how do we put it in context? So first, I'm going to tell you how we go about mapping and characterizing this non-coding landscape. Number two, how we can use this non-coding landscape, these reference epigenome maps, across different cell types to understand what are the tissues where these genetic variants are acting. Number three, we're not going to just look at reference epigenomes, we're going to look at differences in the epigenome. So epigenetic variation in the context of genetic variation, in the context of disease, to basically look at the covariation in this long path to disease. And number 4, how we can use all that knowledge to now go back to that very first slide of this FTO locus and figure out how we can understand the cell type, the target gene, the causal SNP, the regulators, and the mechanism. And lastly, how do we deal with heterogeneity in disease? We like to think of Alzheimer's as a single disease, of obesity as a single disease. We give them names and we hold on to these names. Turns out they're extremely complex and multifaceted disorders. Every patient is affected using a different subset of a huge number of potential etiologies. So we've developed a lot of technologies for addressing that heterogeneity. We're looking at massively parallel reporter assays to basically disrupt tens of thousands of genetic elements at once. We're looking at modeling of electronic health records and multiple traits simultaneously to understand the classification of patients and how they ultimately teach us about disease classification and even the very definition of disease. Who's psyched? Oh, let's try that again. If you guys are not psyched, I'm leaving right now. 3, 2, 1. Who's psyched? Yay! Awesome. All right. So number 1, understanding the epigenome. So this is an incredible collaboration, the Roadmap Epigenomics project. It involves folks like John Stamm, Alex Milosavljevic, Alex Meisner, Brad Bernstein, Joe Costello, Joe Ecker, just an incredible group of individuals, Martin Hurst. And we basically together gathered as a group to understand this fundamental problem of how does a single genetic code give rise to this huge diversity of cell types in the human body. Every one of these cell types has largely the same code, and yet they play completely different functions. So how's that possible? That's possible through these epigenomic marks, which basically serve as memory of the cellular identity of every cell type. So even though they have the same code, that code is modulated, and when they receive maybe the same stimulus from the blood or something like that, they respond very differently depending on the cell type that they are. They modulate the utilization of the genome, and they lead to activation, repression, and so on and so forth. So what is this epigenome that I keep talking about? It's basically the set of all modifications of DNA and its packaging. So DNA can be directly modified, such as CPG dinucleotide methylation. So you have methyl Cs, which is known as the 5th base of the DNA, which basically occurs directly on the DNA. Then you have DNA accessibility, which basically tells you whether that DNA is reachable by the regulators that control it, or whether it's tightly packaged and it pushes all these regulators away. And then the third and by far the richest epigenomic modification are the post-translational modifications in the tails of histone proteins that make up nucleosomes that DNA is wrapped around. So we have 147 nucleotides wrapped around every nucleosome. Every nucleosome contains 8 histone proteins, and these guys have long amino acid tails that stick out. And you can use those to basically add marks, such as, I'm going to put 3 methyl groups in the 36th lysine of histone H3, and so on and so forth. And these basically serve as post-it notes. Kind of like if you have a book of life with 23 chromosomes and 23 volumes, and every person on the planet, every cell type in your body, has the same book of all human knowledge. But some people are plumbers, and others are electricians, and others are physicists, and others are artists, and we use a different subset of that human knowledge. How? By basically putting post-it notes in the right page of the right chapter so that we can very quickly go to the corresponding page and know that there's something important there. So that's what the epigenome does. It basically puts post-it notes in different regions to basically remember where the important parts are. And we can basically read these post-it notes systematically in order to then infer what was the genome doing in every one of these cell types through the state of the epigenome. And there are hundreds of these modifications. And their combinations define different classes of regulatory elements. Proximal promoter regions are defined by a specific combination, and they are the places where polymerase binds and then makes an RNA out of DNA to ultimately make a protein and carry out the functions of the cell. Enhancer regions are much, much further from the place where polymerase binds on the DNA. But they loop around in 3 dimensions, and they bring together a host of other regulators that are bound there that interact with factors at the promoter regions to then assemble the chromatin in a complex that then facilitates transcription and expression of the genes. Other regions are repressed, and these have very specific markings. Other regions are transcribed, and these have very specific markings. So the specific combinations of these marks define different classes of elements. Who's 100% with me so far? Raise your hands. Awesome, great. So we basically set out to map this epigenome across more than 100 different tissues and cell types of the human body, across adult, embryonic stages of development, across different regions of the brain, different types of primary cells, iPS cells, ES cells, in vitro differentiated cells. And for each of those, we basically mapped many different histone modifications, DNA accessibility, and DNA methylation, as well as the quote-unquote output of RNA expression. How do we do that? We use assays like chromatin immunoprecipitation, which basically builds antibodies across, in animal models that basically go and pull down every region that has a particular modification, and then you chop up all the regions that got pulled down, and then you sequence them, and you map them onto the genome. And this is what you end up with. You end up with a density plot that basically tells you how often did each one of these modifications land in this particular region of the genome based on the sequencing reads that were obtained that then match that region of the genome. So you end up with a profile that basically tells you that this particular region had this modification and that modification and so on and so forth. And we end up with dozens of these modifications for each cell type. So we can try to make sense of that data, which is very, very hard, because it's the combinations that matter. And we don't know a priori what these combinations are. So we want to develop methods that systematically learn these combinations and systematically infer the language of the epigenome. And computer science gives us such tools. For example, a multivariate hidden Markov model allows you to look at the observed combinations of individual marks and their combinations and infer the hidden state of the chromatin that gave rise to those modifications. So what that basically allows you to do is separate the observables of the world, which are the individual modifications and where they are, and a model of what's going on underneath, and effectively learn different states of the chromatin that capture different subsets of the genome. And for example, less than 1% of the human genome basically corresponds to these active promoter states. About 3% correspond to these active enhancer states. About 70% correspond to these quiescent states. And that's in any one cell type. As you start combining multiple cell types, a different subset will be active, and that fraction will go down and down and down as you map more and more and more cell types. Who's with me so far? Raise your hands. Awesome. Great. So that basically means that we can now go and systematically map these chromatin states and give them numbers first, and then we can study their enrichments and their conservation patterns and their association with other data sets, and then we can give them names. So then we can go back to the same region which had all of these different histone modification marks and start painting on top of that a single track that summarizes the information, that cell type, based on a set of chromatin states. And here I'm going to be color coding them, as I have throughout the talk, into enhancers in orange, promoters in red, transcribed regions in green, repressed regions in gray. Who's with me so far? Great. So now this is for one cell type. We'd like to do that for 100 cell types. So now I'm only going to show you the stop track for each one of 127 cell types. Are you guys ready for this? This is what it looks like. That's the most dynamic view of the human epigenome that you'll probably ever see, at least for a while. It basically tells you how every region and every nucleotide, every interval, every nucleosome in this region behaves across different tissues and cell types. So in the previous slide, I showed you IMR90 lung fibroblast, and this is, here you can see that the PAX5 gene was in fact repressed there. And the PAX5 gene you can now see is repressed throughout most of the tissues that we looked at, except for a handful where it becomes active. And here's what's really, really cool. When it becomes active, this region here appears to turn on at the same time. Do you guys see that? So that basically means that if I have a genetic variant here, I'm not going to simply say, oh, it's probably one of these genes. I'm going to say, wait a minute, there's a gene that co-varies with that region. Maybe that's the true target gene, and that's a long ways away. Okay? So the beauty of looking at so many different cell types together is not just that we have, 127 times more data than a single cell type. It's also that we have dynamic information, and dynamic information teaches us about the circuitry of the genome. Who's with me so far? Awesome. So that's one view for how to link together these control regions to their target genes. We can basically look at the co-activation patterns of both the regions and the genes. There's another way, which is to look at the physical folding of the genome in 3 dimensions. You can basically look at the same region of FTO that I showed you on the first slide. And you can see FTO sits here, but it actually forms these long-range correlations in this 3-dimensional location with many genes across 2 million nucleotides. That's pretty mind-boggling. It basically means that there's a module here, that these genes all go up or down together because they're looped together in 3 dimensions. So that's the physical linking of the genome, the functional linking of the genome, and then there's the genetic linking of the genome. In the same way that GWAS, genome-wide association studies, look at how genetic variation correlates with case or control for a disease, we can use the same to basically look at how genetic variation correlates with expression levels of a nearby gene. And if this location, every time it has a T, this gene is more highly expressed, that basically means that genetic variation here controls that gene, and we can actually put a link between this non-coding region and the expression of that gene. So there's 3 lines of evidence, physical, functional, and genetic evidence, that allows us to now start linking together these elements. And that's looking downstream. That's looking at who are these regions controlling. What about who's controlling these regions? Well, that's where regulatory genomics comes in. You can actually start looking at the regulatory motifs, the sequence patterns that are recognized by different transcription factors, and how these are enriched in specific tissues and combinations of tissues. So what that allows you to do is basically say that if I see, for example, HNF1B or HNF4A inside an enhancer active in adult liver, I'm going to make the prediction that a mutation in these motifs is more likely to be causal because it disrupts one of those driver motifs that's enriched in the corresponding enhancer set. So we can use all that information to now go back into these genetic regions of association, find all of the genetic variants, and for every one of those, basically say, what are the enhancers that it overlaps? What cell types are these enhancers active in? Who are the regulators that are predicted to control these enhancer regions? And what are the genes that are predicted to be downstream of these control regions? And depending on whether this is the causal nucleotide or that's the causal nucleotide, I have a different set of regulators, and I also have a different set of target genes that I infer from these models. And that's where that complexity comes in. These circuits are extremely distinct from one nucleotide to the other. So we have to be very cautious to find the right target gene, the right cell type, the right regulator, and the right causal nucleotide. Who's with me so far? So we can basically give all that information to the world. How are we going to do that? We built a database called HaploReg for haplotype regulation that allows you to now type the RSID of any SNP in the genome. And it basically tells you, well, here's all the associations of that SNP with disease. Here's the frequency of that SNP across of the different alleles in the different populations. Here's the specific C2G mutation. And now here's all of the SNPs that are co-inherited with that SNP, that are basically descending with that across the human population. So when you have that SNP, you can't think of that SNP in isolation. You have to think of the haplotype block, hence the name HaploReg. So for every one of those SNPs now, we can basically start looking at whether they're conserved or not, whether they fall in enhancer or promoter regions, and what cell types these act in, whether they overlap DNA's accessible regions, or whether they're bound by proteins, and whether the specific SNP in question disrupts a motif by changing the sequence specificity of that motif significantly based on that specific SNP. And of course, whether it overlaps a gene and so on and so forth. Okay? So we have had millions of people downloading this database and using it across industry, academia, pharma, hospitals to basically start interpreting that information. And we could have basically stopped there and said, great, the world has it, we're done, let's go build another database. But in computer science, we like to say that if you eat your own dog food will taste a lot better. So we basically said, let's eat our own dog food here. Let's basically use that information ourselves to see if we can gain any insights into disease. And the first thing we did is basically say, well, let's compare different genetic traits. And let's look at all of the genetic variants that are associated with height. And they paint many different regions of the human genome with LD blocks and SNPs within these LD blocks. And if you look at type 1 diabetes, blood pressure, cholesterol, other traits, you basically find that they paint a different subset of the genome. And that's where it gets kind of cool, because you can basically use these traits and compare them to each other to basically look at their differential enrichment in different cell types, in the enhancers that are active differentially in different tissues and cell types. So when we basically overlay that information with enhancers active in stem cells or immune cells or in heart or liver, what we're basically finding is that different genetic traits are enriched in enhancers acting in different cell types. And we end up with kind of a diagonal that basically tells us that genetic variants associated with height overlap preferentially enhancers acting in stem cells. And genetic variants associated with type 1 diabetes in immune cells. Genetic variants associated with blood pressure overlap enhancers acting in the left ventricle of the heart. And genetic variants associated with cholesterol acting enhancers active in the liver. And this is actually real data. It looks like a cartoon, but it's just a subset of this matrix, which basically looks at 58 different traits across the 127 cell types that we looked at before, and where the enrichments are happening. Here's embryonic stem cells that only light up for height, which is kind of cool, right? Basically says that, you know, enhancers that are acting in early development are basically the place where you want to intervene if you want to affect somebody's height. Or enhancers acting in immune cells is where you want to intervene if you want to deal with, you know, immune traits like allergy, multiple sclerosis, type 1 diabetes. If you want to focus on blood pressure, then you basically look at, bingo, right there, the left ventricle of the heart. And if you want to focus on fasting glucose, basically look right there at pancreatic islets. So this is a completely systematic and unbiased way to basically infer the tissues that are relevant in disease without ever looking at the genes. This is just looking at the non-coding landscape. Who's with me so far? Awesome. Great. So if you look at cholesterol, you basically get liver, huge enrichment, depending on, you know, in regards to what type of cholesterol you look at. If you look at inflammatory bowel disease, inflammatory, that's inflammation, immune processes. Great. T cells and B cells make a lot of sense. Bowel, digestive organs. Again, inflammatory bowel disease has both a digestive component and an immune component. This is great, right? What about Alzheimer's disease? Where would you expect that? Brain. So we have all these different regions of the brain. We look down here, no enrichment for Alzheimer's. And that's a problem. Either our data is wrong, or our method is wrong, or our textbook is wrong. What could it be? Turns out it's none of the above. It's basically that brain samples, when you look at the whole tissue, are made out of neurons, oligodendrocytes, astrocytes, microglia, many different cell types. And they don't show an enrichment, which is puzzling. But what's really kind of cool and satisfying is that we do find an enrichment for CD14 plus cells. These are monocytes, which can be a mark of macrophages and also microglia. Macrophages basically circulate in your blood. Microglia stay inside your brain. And they are the immune cells that are resident inside the brain. They are the ones that are responsible for chopping up neuronal connections that are not being utilized, or for cleaning up debris that accumulates inside the neurons when you use them a lot, and so on and so forth. So that basically says that it's probably not the neurons. It's instead the microglia that play a causal role in Alzheimer's disease. A lot of Alzheimer's research has, of course, focused on amyloid beta, tau, all of these pathologies of the neurons. But this completely unbiased view basically tells us that perhaps we should be focusing on the causality that actually sits upstream of that, and that might actually be sitting along with immune processes rather than neurons. So if you want to fix the cause rather than the consequence of the disease, you have to go to where the genetics is pointing and basically start focusing on microglia. So to do that, we basically collaborated with Li-Hui Tsai, the director of the Picard Institute across the street, and we basically said, let's look at the epigenomic changes that are happening during progression of Alzheimer's disease. Of course, we can't do that in human because humans like to hold on to their brains until they're done using them. So we basically looked at mice who can actually develop symptoms very similar to Alzheimer's. And in fact, Li-Hui's lab has developed this CKP25 mouse that has both early and late symptoms of Alzheimer's and develops them very, very rapidly, within 2 weeks for early symptoms, within 6 weeks for late symptoms, which is remarkable. And now we can look in a temporal fashion, what are the changes that are happening? And we see two types of changes. We see activation of immune processes and then repression of neuronal processes. But temporally, the repression of neuronal processes doesn't happen until later. Whereas early on, you see this activation of immune processes. So that basically says that the changes that are happening in the immune cells precede any change in the neurons. It's not that inflammation is a result of the neurodegeneration and the neuronal loss. It's the other way around. The immune changes precede the neuronal changes. So that basically shifts the therapeutic focus on inflammation as a causal component of Alzheimer's disease. And we can do that systematically for many different traits. If we basically build these machine learning models that allow us to now look across the genome, figure out what are the epigenomic annotations that are the most enriched in every trait, we can then go back to the genetic data and start prioritizing causal SNPs based on the agreement between the genetic evidence and then the prior, which is derived empirically from the enrichment of each of these classes of annotations across the genome. So we can now start saying for every region of association, instead of having all of these possible SNPs, we can basically say, well, what does the genome-wide enrichment tell us? For Crohn's disease, basically tells us focus on immune cells. And in this region, focusing on immune cells starts prioritizing only a subset of those SNPs. For schizophrenia, the epigenomic enrichments basically tell us focus on central nervous system. And in this region, it basically goes from all of these potential causal SNPs into just a handful that we can now start systematically perturbing. And then these SNPs are much more enriched than GWAS SNPs for all kinds of functions like evolutionary conservation or EQTLs or digital genome footprints and so on and so forth. So we can go in individual loci and start prioritizing the SNPs that are more likely to be causal. But we can do something else as well. We can actually start discovering novel loci. We can basically look back at these Manhattan plots. And what you can see is that many people focus on the genome-wide significant SNPs, and rightfully so, because there are so many, we don't even know what to do with them, let alone the non-significant SNPs that are sort of sitting in this twilight zone here. But we basically said, well, human geneticists seek to build larger and larger and larger cohorts. But sometimes you'll run out of patients because simply it's a rare disorder or you run out of funding or something. What if we could combine genetic information and epigenetic information together to prioritize these sub-threshold loci? So Xin-Chen Wang, in collaboration with Lori Boyer, basically built a model that allows us to now start asking, what are the epigenomic features that distinguish genome-wide significant SNPs from other locations in the genome? And can we use these features to now look inside that red box at the non-significant, between 10 to minus 8, 10 to minus 4, non-significant but, sort of tantalizing genetic variants, and prioritize those that are more likely to stay real when we look at larger cohorts. And then he basically looked at a dozen of those that are sometimes 1,000 times less likely than genome-wide significance. And he basically said, let's actually start validating them. And what's really remarkable is that if you look at the downstream target genes and you hit them with morpholinos in a zebrafish embryo, you basically see that they indeed change the phenotype. In this particular case, the heart repolarization interval is actually altered by perturbation of these SNPs that are sitting in, subthreshold loci. And if you look at mouse phenotypes, these genes, if you knock them out, are much more likely to show cardiac conduction or contractility phenotypes than by chance. And if you look at other human-related traits, again, they're much more likely to be associated there. So that basically means that we can actually start prioritizing additional genes and basically expand the set of targets, not just from the genome-wide significant loci, but to many more loci across the genome. And all of that we've done with just reference epigenome maps, just building a map for brain regardless of the person. But here's the deal. Your brain epigenome changes depending on whether you have Alzheimer's or not, depending on whether you're male or female, depending on whether you're 60 years old or 10 years old, and so on and so forth. So these dramatic differences in conditions across individuals were not captured in a reference map. So let's now start expanding these maps across individuals. So we're going to look at both genetic and epigenetic information jointly. And this is trying to span, to basically bridge this very large span between genetic variation and disease by basically looking at the specific tissues where a variant is acting, the regulatory regions that are altered, the expression patterns of nearby genes, the intermediate phenotypes, all in the context of disease, so that we can start painting a path for how we can get to, say, Alzheimer's disease through all these intermediates. The challenge, of course, is that the environment can affect the disease and these intermediate variables, so these arrows are not unidirectional in the middle here, they're actually bidirectional. And number two, there's a lot of feedback from the disease itself that might be influencing those. So we have to be very cautious when interpreting these in terms of causality, and we can develop tools to overcome this. So we tried this approach in the context of an incredible collaboration with David Bennett from Rush University and Phil de Jager, who used to be here at the Harvard Medical School and who's now in Columbia, in the context of the ROS-MAP study, the Religious Order Study and Memory in Aging Project. So this is a cohort of more than 1,000 individuals that David has assembled over decades, who basically agreed to donate their brain to science post-mortem and who agreed to be subjected to cognitive evaluations over more than a decade. Donated blood, they have stored samples across, this incredible wealth of data. And what we basically looked at is the correlation between genetic variation for those individuals, epigenetic variation in these individuals, and phenotypic variation in these individuals. And we did that by focusing on the dorsolateral prefrontal cortex, a central place of cognition. The first thing we found is that there's a dramatic correlation between genetic variation and epigenetic variation. We find 50,000 methylation QTLs. These are basically genetic variants where if I tell you that I have a TT, a TG, or a GG, I can basically predict the level of methylation for that nearby CPG. And that's pretty remarkable. It basically says that we can actually start making predictions about brain methylation simply by looking at the genotype. And that's very useful because for those people who chose not to donate their brain before they're dead, which is everyone, hopefully, we basically can say, well, I don't have access to your brain, but I'm guessing that your methylation pattern is as such based on your genotype because we've done this across these individuals. And that's where it becomes really, really powerful because I can basically say, instead of doing a genome-wide association study, I'm going to do a methylome-wide association study, an MWAS, to basically look at how methylation varies with disease. But an MWAS, first of all, can only be done in few individuals, the ones that actually donated their brain, and only gives us bidirectional arrows. And that's because some of that methylation variation might be a consequence of the disease. But that's where the methylation QTLs come in, the methylation quantitative trait loci come in. Because I can basically say, if I can predict methylation from genetics, why don't I do an MWAS with predicted methylation? So I'm going to do an imputed MWAS. So I'm going to predict for every individual the level of methylation, and I'm going to use that level of methylation to then correlate methylation with disease. And here's where it becomes really important to have predicted methylation. Number one, it allows us to now look at many more individuals. Number two, it allows us to look at only the genetic component of methylation, only the stuff that I can actually predict from genetics, which is very important because suddenly that arrow becomes unidirectional. Who's with me on this one? Raise your hands. Awesome. Lovely. So we can do that if we had genotypes of these individuals, but if we don't, we can actually do that at the summary statistics level, a method pioneered by Gusev et al., known as TWAS. We can apply that in the context of methylation and basically start predicting the level of methylation of tens of thousands of probes across the genome based on the genetic information by combining genetics from multiple loci together to predict methylation. And if you look at cross chromosome 6, genome-wide significance would be above the top of the slide, and there's just no loci that are genome-wide significant. But if you do this approach of basically going through the methylation, you basically find that there are half a dozen significant loci by combining multiple SNPs. And it's very powerful. Number one, it gives us new genetic loci. That's a big deal. Number two, it actually tells us that these genetic loci likely act through methylation in the brain, because that's the tissue, the stage of development, and the type of regulatory activity that we're looking at. So they predict the region, they predict the cell type, they predict the something more about the mechanism than simply, oh, there's a genetic correlation. So this is extremely powerful, and it allows us to now start expanding this to look at causal paths from genetics to methylation to transcription to disease, expanding beyond just a single step. We can use surrogate tissues such as blood to basically improve our prediction of these intermediate variables. And we can actually start doing that across many different tissues. So just a couple of weeks ago, the GTEx Consortium just published this largest QTL study across 44 different human tissues. And we can actually start combining information across multiple tissues. You can basically see that many SNPs are in fact active in many tissues simultaneously, and there's this huge correlation pattern where QTLs in brain are very similar depending on, regardless of what brain region you're looking at. So we can actually start improving our power by combining prediction across multiple tissues and regions of the brain. For example, if you look at the number of samples in different tissues in GTEx, you basically see that the samples that have the fewest, the tissues that have the fewest samples also have the fewest EQTL genes. So that basically means that our power is dramatically reduced for those tissues with few samples. However, when you start combining information from multiple tissues jointly by learning these factorized models with projection of these genotype by expression matrix into a lower dimensional space, you can actually leverage information from many different tissues together and basically start discovering many, many more QTLs. So applying that in the context of Alzheimer's basically allows us to now start finding brain QTLs that are extremely significant for several different regions and genes. And what we're finding is that for even subthreshold genes, we can actually, subthreshold genetic loci, we can find new genes that are very strongly correlated with the disease status. So that basically means that we can now start expanding the set of loci, start gaining insight as to the potential mechanisms in these loci. And I wanted to now switch to one such example where we try to uncover these circuits from the genetic variant all the way to the phenotype through all of these intermediate steps. Namely, to start with the region of association, find the relevant tissue, the target genes, the causal nucleotides, the upstream regulators, the cellular phenotypes, and the organismal phenotypes. And we're going to do that in the very first slide again on the FTO locus, and this is an incredible collaboration with Melina Clausenter, who's now a professor here at the Harvard Medical School. You know that obesity is extremely important. It costs more than $200 billion a year in the US alone. It affects 500 million people worldwide, and it's usually thought of as a behavioral problem. You should eat less, and you should exercise more. But in 2007, the first genetic association with obesity was discovered, basically suggesting that maybe there's something in our genes that's stronger than our willpower. And this genetic association, as I showed you, sits in this FTO gene. But it doesn't necessarily act through FTO. So let's go through the steps of basically figuring out where could it possibly be acting. A lot of people thought that it would act in the brain to basically modulate motivation or exercise or food choice. Our epigenome data suggests that in fact it acts in mesenchymal stem cells. These are the precursor cells that give rise to either your white fat cells or your beige fat cells. And your beige fat cells are basically your best friend. They are the places where you can burn excess calories simply by activating your mitochondria and dissipating heat. So you can basically burn calories rather than store calories. White fat cells are your worst enemy. They are the places that store all that excess energy into lipids for a rainy day. Of course, these used to be great when we had little food, and these used to be terrible. But now the situation has flipped because we have many more calories in our diet than we have exercise to expend them with. So this is where the variant may be acting. But what are the target genes? Well, as I told you earlier, this genetic association localizes squarely in the FTO gene. But there's this high-C information of physical linking that implicates potentially many more genes nearby. So with Melina, we basically looked at all of these genes and their expression differences across risk individuals and non-risk individuals. And it turns out that if you have the risk variant, you show twofold higher expression of two target genes. And these are sitting 1.2 million nucleotides away and 500,000 nucleotides away. FTO does not show any difference in expression here. And what's really cool is that this is actually a gain of function. We always think of losing the activity of a gene. In this particular case, the genes become hyperactive. And we narrowed down the association to a single nucleotide that disrupts this AT-rich interacting domain by changing a T into a C, and that changes the binding of this ARID5B repressor that no longer can bind the DNA motif, no longer can repress enhancer activity, leading to a de-repression of IRX2 and RX5, this twofold increase in expression. What do these genes do? Well, they're negatively correlated with mitochondrial activity, and they're positively correlated with lipid metabolism, suggesting indeed a shift from energy dissipation to energy storage in the risk individuals. And indeed, if you look at the adipocyte size, you find that they have much larger adipocytes, if the individuals that contain the risk variant, and they also have much less activity at the mitochondrial level, suggesting indeed that this model is real. So that's our model. Our model basically says that ARID5B normally binds this motif and represses these guys, but when that single SNP happens, these IRX3 and RX5 target genes get de-repressed and then shut off the thermogenesis process, shifting to white fat cells and storage of lipids, and shifting from lean to obese. What's really important about having a circuitry is that you can actually start manipulating that circuitry. You can actually start knocking down or overexpressing the genes implicated, either as target genes or as regulators. You can actually start editing the single nucleotide variant that's predicted to be causal in this particular case, using CRISPR-Cas9 genome editing. So the results for all of these modifications are remarkable. They're kind of like a switch between lean and obese phenotypes. If you actually edit that single nucleotide, out of 3 billion nucleotides, you pick one and you go and edit it, you basically see that cells from risk individuals who are unable to carry out thermogenesis, who are unable to burn heat, suddenly have that process completely restored by a factor of 7. If you look at mice and you take the downstream target gene, IRX3, and you knock it down in adipose tissue, you basically see that the mice lose 50% of their weight. All of their white fat stores are completely depleted. And if you look at what happens in a high-fat diet, you introduce a high-fat diet, normal mice are heavy and they get heavier. These lean mice don't gain any weight in a high-fat diet. That basically means that they're simply burning all these calories. They don't change their appetite. They don't change their exercise levels. Instead, they're burning more energy when they're awake, and they're burning more energy when they're sleeping. So that basically means that by manipulating the circuits that come out straight from the genetics, not just in the individuals who have a genetic variant, but in anyone in mice, in human, we can actually start changing these circuits as engineers and manipulating them and turning those same knobs that nature turns only a little bit by a lot, by changing the same circuitry. We want to do that systematically, and we want to do that at a large scale. And we have to deal with a huge heterogeneity of disease, where many different individuals have different genetic variants implicated in disease. So we've developed methods for systematically perturbing these variants across 10,000 at a time using massively parallel reporter assays, and basically being able to narrow down the regions of association to specific nucleotides using these episomal high-throughput assays. And we're able to now differentiate stem cells into different cell types to basically see where these variants manifest themselves. And in collaboration with Kevin Egan, we're now looking into high-throughput perturbation of cellular phenotypes, 384 wells at a time, to basically look at synaptic activity and neuronal activity in the context of schizophrenia. And in collaboration with several labs, we're now looking at the combination of multiple phenotypes. So in the case of Alzheimer's, we have neuritic plaques, neurofibrillary tangles, neuroinflammation, but not every individual who's marked as Alzheimer's has all three of them. Every person has a different subset. And what's really cool is that if you look at different subsets of phenotypes, they are associated with epigenomic changes in different regions of the genome. And these regions of the genome are acting in different cell types. And they are associated with genes involved in different functions. So what that basically says is that we can actually start taking these monolithic diseases and break them apart into their components. And we've started doing that systematically across electronic health records for millions of individuals to basically look at the comorbidity patterns across a large number of diseases. And that completely changes your view of disease. Instead of basically thinking of Alzheimer's or schizophrenia or bipolar as separate entities, we can basically start defining metaphenotypes. And using those metaphenotypes, we can actually start doing a lot of things. We can start imputing additional phenotypes that are not measured for a particular person. We can start imputing lab tests and basically say, oh, you should probably take that test because I predict it's going to be positive. We can start carrying out genotypic associations across much larger sets of individuals and with much more biologically relevant phenotypes because we're looking at the metaphenotypes rather than these human-defined phenotypes. So that's what I want to leave you with, the fact that we can actually start systematically studying the human epigenome, use that to identify disease-relevant tissues, variants, and regulators, look at not only genetic variation across individuals, but combine both genetic and epigenetic variation, start using that to uncover and then manipulate the disease mechanism and circuitry in specific loci by learning across the genome, and then applying that to individual loci. And we can start tackling disease heterogeneity using massively parallel reporter assays and modeling multiple traits simultaneously, all by looking at the central integrator of the epigenome. So we're now able to move much closer to this manipulation and ultimately therapeutic development, just as industry is actually moving closer to the discovery and building these resources. So we've established a lot of new kinds of collaboration, bridging academia and industry to basically look at new models for discovery. And this is an extremely exciting place and time to be doing all this. So this has been an incredible collaboration with many individuals. I mentioned the high-throughput assays, the electronic health record collaborations, the FTO collaboration, the Alzheimer's collaboration, the ROSMAP collaboration, and of course the Roadmap Epigenomics project. And I'll stop there and let you guys continue. Thank you.